Come on. I forgot to get that recorded. If you don't see a button showing that I'm recording, please remind me that I need to do the recording. I don't see the button when I do the screen share, meaning I don't see anything. So if I'm not recording, then other students can't get it. So please let me know if I'm not recording. All right, as I stated, I always start with a slide showing you the major goals and the rough outline. Please know the terms. We'll talk about each of these, or you'll do it in the in-class assignment. Coccus, Bacillus, Spirilla, Strepto like Streptococcus, Staphylo like Staphylococcus, Diplo like Diplococcus, Tetrad, Sarcenia, Vibrio, Spirochete, Glycocalyx, that's a part of the cell. It's actually external to the cell. Flagella, axial filaments, fimbriae, pili, peptidoglycan, which I think we've talked about that a little bit, but we'll talk about it in greater detail today. Endotoxin, selective permeability, diffusion, facilitated diffusion, osmosis, active transport, tonicity, plasmid inclusions, endospores, and very last topic I'll talk about, not today, probably tomorrow, the endosymbiotic theory. So today we're going to talk about how gram-positive and gram-negative cell walls differ. And we did talk slightly about that, but we'll go into greater detail. And also a major goal is to describe the importance of taxa in bacteria. Any questions about any of that? If not, let's move on. Get it to move. There we go. So you should know the difference between prokaryotes and eukaryotes. A prokaryote means pre-nucleus, Greek for pre, and literally that's kernel, but it's translated into nucleus. And eukaryotes, Greek for eu, meaning true, so true nucleus. The prokaryotic cells have one circular chromosome. They're not in a nucleus. They don't have a nuclei. They don't have organelles either. That's down there. And the eukaryotes, they have generally linear chromosomes, multiple linear chromosomes, and the chromosomes are in a nucleus. They also have organelles. And I should mention, when I'm saying the word organelle, I mean membrane-bound organelle. The DNA of prokaryotes, well, at least of the bacteria, they do have proteins on their DNA, but they do not have histone proteins on the DNA. In the eukaryotes, the DNA is wrapped up on histone proteins. The bacteria, and I'm only talking about the bacteria here, have peptidoglycan in their cell walls. The archaea, most of them do not have peptidoglycan. And the eukaryotes have no peptidoglycan in their cell walls. And as you know, animal cells don't even have cell walls. The prokaryotic cells, most of them divide by binary fission. That's where the cell splits into two. Eukaryotes, their cells divide by mitosis. Any question about any of that? There are also differences between the prokaryotes and the eukaryotes in their size. The prokaryotes, their sizes range from about, and obviously this is an average, 1 micrometer to about 10 micrometers in length. And this 10 is how long they are. The prokaryotes, some of them are much longer than they are wide. So their diameter doesn't go up to 10 micrometers. In the eukaryotes, they're larger. On average, they start from about 10 micrometers and they get larger than that. I mean, the largest cell you've seen is the chicken yolk, which is much larger than most cells. Most eukaryotes go up to about 500 micrometers in length. The prokaryote shapes can differ. They have three basic shapes, and it's true they can have other shapes, but most of the other shapes are some variation of these three basic shapes. There can be round cells, and we call those coccus if we're talking about a single cell. If we're talking about plural, it would be cocci. Bacillus is a single cell looking like a rod, and that's a single cell. If you're talking about plural mini cells, we'd say they're bacilli, and you can call them rods or a rod. And then the other cells are spiral cells, and that's singular. If they're plural, it would be spirilla. Any questions about the basic shapes? There are different types of spirals, and you can argue, is a vibrio a rod or a spiral? We're not going to get into that argument. In this slide, they're listed as a spiral here and then a curved rod there. So even that's trying to avoid the argument. And they are a rod, but they're bent or curved. There's also the type of spiral, the spirillum, and it's a simple spiral with one or a few curves. They're more rigid. They have a corkscrew shape, and you can see this spirillum has two bends to it. And then there's cells with many bends, and they are flexible, so they're less rigid than the spirals, and we call them spirochetes. Now, you don't need to memorize that this is a spirochete and that is a spirillum, because I'm never going to ask you to differentiate that. But these are the terms for these cells and for this cell, and we just call them all spirals. Okay? The prokaryotes do come in some unusual shapes, and you're seeing unusual shapes here. There are some that are star-shaped, like the Stella bacteria, kind of look like a star. And then there's some that are actually square. This is an archaea, the halo arcula, and there's an actual, which I guess I shouldn't say square, they're rectangular. An actual electron microscopic image of the cell. And most bacteria, even if they're unusual, they come in a monomorphic shape, meaning that the cells have one shape, like all of these are kind of rectangular, all of those are kind of like a star. But not all bacterial species are monomorphic. A few are pleomorphic, and pleomorphic means they have many shapes. And let me go ahead and blow that up just for a little bit better. This is mycoplasma, and as you can see, they have many different shapes. Pleomorphic. Any questions about that? Yep, I didn't in the slideshow. There we go. When we're talking about prokaryotic cells, we can talk about their cellular arrangements, sometimes also called growth arrangement or cellular growth arrangement. And that is how the cells grow. What you're seeing with the cellular arrangement is that there will be more than one cell sticking to each other. And that's just because their cell walls, after they divide, stick together. Now, you don't always have to have a cellular arrangement, and some cells, especially the spirals, their cellular arrangement is always uh, unicellular, meaning one cell. You only find them and see them as one cell. But sometimes you can find them with a cellular arrangement where there will be diplococci, two cocci together, or a bunch of them strung together in a row, which we call streptococci. This has to do with the plane on which the cell divides. If it only divides on one plane, like this here, then it'll grow as a diplococci or a streptococci. If they grow on multiple planes, they can form, the cocci can form a tetrad, and this divides this way, and then the next division will be this way, and that will form a tetrad, which is a group of 4 cells. And that one looks like it's starting to go through division, but there's a group of 4 easily seen there. If they further split on a third plane, meaning they grow in 3 dimensions, you can get a sarcenia, and that'll be a group of 8 cells. My mouse died. 1, 2, 3, and then 4 behind it. 1, 2, 3, 4, 8. And it's kind of hard to see, but if you were to count that group there, that would be a sarcenia, a group of 8 cells. You can also, when you divide in all 3 planes, you don't have to form a sarcenia. You can also have a pile of cells, and we call that a cluster. And if it's cocci and it looks like that, we call it staphylococci. Staphylococci means that it looks like a cluster of grapes. Any question about that? And that would be a staphylococci. Now, for the cellular growth arrangements to see these, you usually have to grow your cells in a liquid culture, because if you grow them on a Petri plate, the cells just all grow together, meaning solid medium on a Petri dish. They just all stick together, and then when you put them on a slide, they're just clumping. So you might not see the cellular growth arrangement if you grow the cells on a Petri dish and then make a slide out of them. To see the cellular growth arrangement, you actually have to grow them in a liquid culture and then put it on the slide. And even then, the cells can break apart. Like you never see only streptococci. You will see streptococci, and then the streptococci breaks, so you'll see some diplococci, and then the diplococci breaks, and you'll see some unicellular cells. Is that clear? Whenever you see cellular growth arrangements, it's pleomorphic. There will be more than one cellular growth arrangement. And when you're calling it, you go the biggest one you see, which in this case would be streptococci, because there will also be some diplococci and some singular cells. Any questions about that? Now, this is the cellular growth arrangement for cocci, and as you can see, it's complex. There's unicellular, diplo, strepto, tetrad, sarcenia, and staphylococci. For bacilli, you can have some of the same cellular growth arrangements, but they have fewer. You can have single bacillus together, meaning they're growing as singlets, and this cell and that cell would be that. You can have diplobacilli, which I don't see any of those there. Maybe, no, that's a triplo right there. I don't see a diplo. Anyways, that would be 2 stuck together. And then you can have streptobacilli, where they're sort of growing like a sausage link. And that would be a streptobacilli right there. There's 3 of them stuck together, you can see. You can also have them not growing like a string of sausages. You can have them, let me end this because that's not showing too well. That's sort of getting into it. You can also have them growing as a string of bacilli, but instead of being connected on the long side, they're connected on the short side. And these we call a palisade. And that's because if you put them on this side, meaning rotate them 90 degrees, so this side is on the bottom, it sort of looks like a palisade fence. And if you don't know that term, it used to be very common, and you could read about palisade fence in, it was either Tom Sawyer or Huckleberry Finn. Whoever wrote those stories wrote about the palisade fence, and one of the boys had to go paint the palisade fence, if you remember, and he tricked the other boy. It was probably Tom Sawyer because he was the smarter one. Tricked Huckleberry Finn into it being a fun job to do, and so he tricked Huckleberry Finn into helping him paint the palisade fence. Anyways, that's where it comes from, palisade, because it looks like a palisade fence. Any question about that? That's the different cellular growth arrangements for for the bacilli. And as I stated, for the cellular growth arrangements, you never see just one type of cell. If they're streptobacilli, you will see some streptobacilli. And if they break into twos, there will be diplobacilli. And if that breaks, there will be a single bacillus. Okay, so you'll see the cellular growth arrangements as pleomorphic. They won't just be one sort. All right, and I don't know why it is in this slide here. It's also showing you that the bacilli, besides being the shape of bacillus, can be a coccobacillus, and that's not a cellular growth arrangement. That's actually the shape of the cell. And what happens if you shorten the bacilli and get it shorter and shorter? When it's very short, it looks something like that. And that's in between a bacilli and a cocci. And so we give that term a coccobacillus. It's a cell that sort of looks both as a bacillus and a cocci. And there are some bacteria that take that shape. Any question about any of that? So that's a shape, not the cellular growth arrangement. Excuse me, trying to move on to the next slide. All right, any question about the cellular growth arrangements or the cell shapes that bacteria take? If not, let's move on to the structure of some typical prokaryotic cells. Here you're seeing most of the structures of the typical prokaryotic cells. You always find the red items in all bacteria, and for the cytoplasm, the plasma membrane, and the DNA, you find those in all cells. So all cells always have DNA, a plasma membrane, and cytoplasm. Actually, eukaryotes also have ribosomes, but their ribosomes differ from prokaryotic cells. You can also find other structures in the cell as shown here, and we'll talk about each of them in inclusion, which would be there. That might be an inclusion as well. You can have a capsule outside the cell wall, and there yellow would be the cell wall. Plasma membrane would be the inner layer of the I don't know, the barrier here. Fimbriae, a plasmid, that would be a small piece of DNA separate from the chromosomal DNA. And you might see flagella. This is multiple. If it's just one, it's flagellum. Let's talk about each of those cellular structures. The glycocalyx, also pronounced glycocalyx, is secreted by some prokaryotes. And yes, there are many prokaryotes, but if we look at the majority of the prokaryotes, they don't have a glycocalyx. So I'm going to say some prokaryotes. And we're really only going to talk about the bacteria, but the archaea also have a glycocalyx. The glycocalyx is a structure that the cell secretes, and it's outside of the cell wall. So we say the structure is external to the cell. It's usually sticky, and there's 3 types of glycocalyxes. There's a capsule, which is neatly organized. It can be seen under the light microscope. It's mostly water, but it does have a significant portion of other molecules like carbohydrates. And the capsule can prevent the cell from being phagocytized, like by a white blood cell. So it can protect the cell by preventing phagocytosis. So the capsule can be a pathogenic structure. Another type of glycocalyx is the slime layer, and it's a less organized structure, meaning loosely organized. It's mostly water, and because of that, you can't see the slime layer with a light microscope. To see the slime layer, you'd have to see it under the electron microscope, or let's just say just with the electron microscope. And the reason being is it's mostly water, so it just doesn't show up with the light microscope. And it's extremely fragile because it's mostly just water. So whenever we're talking about a glycocalyx on your bacteria, we'll usually be talking about the capsule because that's the one you can see with a light microscope, and that's all we will use, and you guys will only be using the microscopes virtually because you're never going to be in a microbiology lab, but you will use it virtually. There's a 3rd type of glycocalyx, and this is totally new. It's the extracellular polysaccharide layer. It wasn't known when I went through with microbiology. It's a glycocalyx in a bacteria that's forming a biofilm. And we'll talk about those later in the term, biofilms. Any question about the 3 types of glycocalyxes? You can also see in bacteria flagella, and the flagella is mostly external to the cell, but there is a portion of the flagella which is hooked into the cell wall and even on the plasma membrane, but most of it is external to the cell, meaning external to the cell wall. So when we're saying something is external to the cell, we're saying it's outside of the cell wall, which is kind of funny because the structure that determines whether something gets inside or outside the cell is really the plasma membrane, but we do consider the cell wall a portion of the cell. Any questions about that? I'm sorry, it's a little complex. Anyways, the flagella, or flagellum if there's only one, is used for locomotion, and if the bacteria can swim, move around by swimming, then it has at least one flagellum. It may have multiple flagella. And the flagella are made of the protein flagellin. Any questions about that? I'm not going to require you to memorize the different types of flagellum or flagella. Here we go. But there are names if there's only one. Like I said, you don't need to know it. There's names if it's on both sides and two, and if it's two on both sides, and then if it's multiple, and I won't ask you to know the names. I'm just showing you that there are different names for the different arrangements of flagella. Any questions about that? All right, so if the cells can move, and I'm talking about swimming, there is another form of movement, and it's debatable even whether it's a form of movement, and that is bacteria on a solid structure can slide, and it's been determined that how they do that is that they're not using the flagella to slide. They're using the fimbriae, which we haven't talked about, and the fimbriae is out of the cell, and then the fimbriae retracts, and as it's retracting, if the fimbriae holds on to the solid media like the agar on a petri dish, and then it retracts, it can pull the cell forward, and then once the cell is moved up, there will be new fimbriae which can go out and attach to the media and pull up. And you don't really need to know, but the point is that cells can also be described as sliding, and it is a form of movement, but I'm not going to hold you to knowing that it's a movement because it's kind of complex. And if we're talking about cells moving, we're talking about them moving because of flagella, and they can actually swim in a liquid media. Did I confuse anyone there? I probably shouldn't have even talked about sliding. Anyways, if cells can move by swimming because they have flagella, meaning they rotate or tumble or run or swim in a liquid media, we call this movement taxis. And taxis is movement toward or away from a stimuli. Like phototaxis, I should say there's two types of taxis. You can have phototaxis, that's movement towards or away from a light. And then there's chemotaxis, and that's movement towards or away from the chemical stimuli. Any question about taxis? Most organisms, if they have phototaxis, they would be photosynthetic, and so they'll want to move towards the light source. But this one, if you notice, its flagella are that side, that way, so it's actually moving away from the light source. Chemotaxis, depending if it's a chemical the cell wants, they'll move towards it. If it's a chemical they don't want, they'll move away from it. And they're moving either towards or away to get in a more favorable environment. Now, cells don't have to move by taxis, meaning they can move randomly, and that is they're not being stimulated to move one way or another. Any question about any of that? Okay. There is another flagella, and we call this an endoflagella or axial filament. It is a flagella, but let me blow this up. The flagella is right here, sort of wrapping around the cell, and the flagella is inside the cell wall. Oh, I can't blow that up. Let me see if I can blow this up a little. Oh, I could, but let me get that out of the way. So here is the cell wall. My mouse stops dying, darn it. There we go. So the cell wall is going around like that. And there you see the axial filament inside the cell wall. So that Endoflagella, this right there, is actually inside the cell wall. And there you see an actual electron microscopic image of the Endoflagella wrapping around the cell. And cells that have endoflagella are only in the spirochetes. The endoflagella is anchored at one side of the cell and then wraps around the cell, but it's inside the cell wall. And it causes the cell to move. Let's see if I can get this to work. Let me try if it works now. Oh, there it goes. When I had it under the expanded menu, we weren't seeing it. It causes the cells to move in a corkscrew-like movement. And hopefully this will load. Well, it's still coming. There we go. And you see how the cell can just change its movement. That even went a 90-degree turn, but the point is it can move back and forward. And it's just moving in a different direction by changing how the endoflagella moves. Like this one will come down, and I think it's going to change someplace down here. Let's see if I can scroll down. Nope. All right, any question about any of that? Anyways, that's the corkscrew-like movement. Here are the fimbriae, which is a structure external to the cell. And the cell generally uses fimbriae only for attachment. They're not for swimming. And the fimbriae are shorter than the flagella. They're also shorter than the pili, which I'll show you in the next slide. So cells use the fimbriae for attaching to surfaces. And some human pathogens use their fimbriae for attaching to our cells. So the fimbriae can be, it doesn't have to be, but it can be a pathogenic structure. All right, any question about the fimbriae? The pili are shown here. And this is a structure external to the cell wall. I guess I should have said that for the fimbriae. That is external to the cell wall, although both of them are anchored in the cell wall. The sex pili is called sex pili because it literally comes out of the gram-negative cell shown here and skewers, that comes out of the F plus cell shown here, and it literally skewers the F minus cell shown here. And then this sex pili acts as a winch to bring the two cells closer together. They do not have to be touching for the sex pilus to work. The sex pilus is hollow, and a plasmid can then leave this F plus cell and then move into the F minus cell. The point is that the sex pili is used to transfer DNA in a plasmid from one cell to another cell. Any questions about any of that? All right. If there's no questions, let's talk about the cell wall in additional detail. The prokaryotic cell wall is complex. It's a semi-rigid structure responsible for the cell shape, meaning the plasma membrane doesn't give the cell a shape because it's fluid, soft, fragile. All of those terms can be used, as well as thin. The prokaryotic cell, if it has a shape, which usually they do, that shape is because of the cell wall giving it that shape. The cell wall, besides being semi-rigid structure and giving the cell a shape, also protects the cell and helps prevent osmotic lysis. So if you have like a red blood cell, which doesn't have a cell wall, and you put it into water or any liquid which is in, let's see, hydrothermia? No, that's not right. Hydroosmosis? Anyway, something like that. Where there's more water outside the cell than inside the cell, water would move into the cell, and water would keep moving into the cell, a red blood cell, until the red blood cell puffed up and got so much water that if it gets more water, the cell membrane bursts and then the cytoplasm leaks out. And that would kill the cell, resulting in osmotic lysis. But if a cell has a cell wall, water will move into the cell until the plasma membrane is pushed up against the cell wall. And then the plasma membrane cannot move any further because the cell wall is preventing it from expanding. And when that happens, no more water can move into the cell, even though there's more water outside the cell than inside the cell. And that prevents a cell with a cell wall from undergoing osmotic lysis. Any question about that? So the cell wall prevents bacteria or prokaryotic cells from osmotic lysis because yes, water will move into the cell, but only to the point where the plasma membrane expands up to the cell wall. The cell wall, of course, is located external to the cell membrane, and the cell wall can contribute to the pathogenicity of some species, meaning that there could be something on the cell wall which could make people sick, and Mycobacterium is an example. It has mycolic acid in the cell wall, which can lead to the pathogenicity of the species. The cell wall, the main molecule in it for bacteria, and we're only talking about bacteria here, is the molecule of Peptidoglycan. Let's talk about Peptidoglycan in more detail. You can find it in bacteria cells, and for this course, we're going to say that's the only place you will find it. Peptidoglycan is made up of a polymer of two repeating disaccharides. The disaccharides being NAG or NAM. Let me blow this up a little bit so you can barely see that, but you can see it. NAG, if you don't want to call it NAG, you can call it N-acetylglucosamine, or NAM, if you don't want to call it that, you can call it N-acetylmuramic acid. And that's what makes up peptidoglycan, NAG and NAM, and there we go, NAG repeated by NAM, repeated by NAG, repeated by NAM, and it just goes that way in the chain. You can see it here, NAM, NAG, NAM, NAG, NAM, NAG. these two disaccharides are linked together, meaning they're hooked together with a covalent bond this way, but they are linked together this way and this way, meaning the chains of disaccharide are linked together with short peptide links. And there's one going up and down, and that would connect this one with that one shown there. And then this one connects that one together, and it's linked here and here, and then it's linked here. So there's a short peptide linking this one to that one. These short peptides and then the disaccharides, the two disaccharides are what give peptidoglycan its name. The disaccharides are glycans, and it's connected with peptides, so it's called peptidoglycan. Any question about that? Penicillin actually inhibits the cell wall by inhibiting the peptide bridges linking the peptidoglycan together. So penicillin, if the cell is making the peptidoglycan in the cell wall, penicillin will get right here and then prevent this peptide from forming and linking this disaccharide chain to that disaccharide chain. So penicillin inhibits the lattice formation of the peptides, which then puts a hole in the cell wall, and the cell membrane can protrude out of that hole. And that can lead to lysis of the cell, meaning if there's a hole in the cell wall, the cell membrane can protrude out of that hole, and then water can enter the cell, and the cell membrane will just keep protruding out of that hole in the cell wall until the cell membrane eventually bursts. And we'll talk a little bit more about that later. Any question about peptidoglycan in the cell wall? We should mention that there are two types of cell walls, and I think I've already talked about that. There are gram-positive bacteria cells, and when we look at it in this picture here, yellow being the cell membrane, the gram-positive cell wall has a very thick cell wall shown in purple. And we look at that cell wall, it's many layers of peptidoglycan, more than just shown. Actually, I think there's only 3 layers shown here, but it's actually many, meaning there's many layers, probably hundreds of layers, maybe even thousands of layers of peptidoglycan on top of each other. And when we look at that cell wall, it's simple compared to the gram-negative cell. There's the plasma membrane, which is a lipid bilayer. It does have proteins in it. And then above that is the cell wall, and it's only the peptidoglycan, meaning the disaccharide, and then the peptide links shown in aqua and green, holding it together, and there's many layers of it. There are a few other molecules in the cell wall, mainly teichoic acid, shown here, and lipoteichoic acid, which is teichoic acid with a lipid end. The lipid teichoic acid, the lipid portion is in the cell membrane. There we go. So this would be the lipid portion of the lipoteichoic acid. The teichoic acid is only in the cell wall. The teichoic acid portion, or the lipid teichoic acid, or the teichoic acid, helps hold the peptidoglycan together. And it may also be responsible for allowing ions to move through the cell wall, meaning the ions may move down the teichoic acid. Any question about that? So that's the gram-positive cells. Let's talk about the gram-negative cells. Whoops, nope, still on the gram-positive cells. Actually, I've already talked about that. I've already talked about that, and that they may help movement of cations. So I've actually already talked about that. Teichoic acid is a polysaccharide. It can provide some antigenic variation within the cell so that this cell will be different from another cell that has antigenic variation in the teichoic acid, just allowing the immune system to respond to different cells. The gram-negative cells of bacteria differ from the gram-positive cells, and when we look at the cell wall in this picture here, it's the same, that there's a lipid bilayer in the cell membrane here, and then we see the peptidoglycan cell wall, and it's there, but it's much thinner than in the gram-positive cell. So the gram-negative cell wall is thinner, especially the peptidoglycan, and then on the outside of the cell wall, we have another lipid layer. And if we look at that in greater detail, there's the cell membrane, which is not part of the cell wall. And it's a lipid bilayer, which does have proteins in it. And then we see the peptidoglycan layer. It's only showing one layer of peptidoglycan. That is not correct. There is more than one layer of peptidoglycan in the cell wall, but there are fewer layers than in the gram-positive cell. So there are several layers, I don't know how many, of peptidoglycan. It might be many, but it's fewer than in the gram-positive cells. Is that clear to everyone? And then on the outside of the peptidoglycan is part of the cell wall, but we call it the outer membrane of the cell wall. And when we look at it, come on, mouse, the lower layer of the outer membrane is largely made up of the same molecules as you have in the cell membrane, meaning it's mostly made up of phospholipids for bacteria. But when we look at the outer layer of the membrane, the bilayer, we see that we have another lipid, which is called lipid A, the big blue one here. And attached to it is the O polysaccharide, making the hairs that you can see here. So that together, this molecule is called a lipopolysaccharide. And this is important because lipid A is an endotoxin. So of the lipopolysaccharide, the lipid A portion is an endotoxin. And endotoxins are always important. Actually, all toxins are important because you never want to be exposed to a toxin. It's generally bad. They're toxic to our cells. That's why we call them toxins. In this outer membrane of the cell wall, there's a special protein, which is called the porin protein, and it allows molecules to leave the outer membrane, meaning this is a lipid bilayer. For molecules to enter the cell of a gram-negative cell, they have to first, to enter the cell, they have to first come through the outer membrane, and they do that through the porin protein channel. And then they have to come through the cell membrane, and there's no porin proteins in the cell membrane. What they do to come through the cell membrane is they come through a transport protein. If they need to go through the lipid, the two bilayers, membrane bilayers of a gram-negative cell, okay? And for a gram-positive cell, they only need to come through the cell membrane. And if they need to get through the cell membrane by going through a protein, there will be a transport protein. And that's for molecules to come into the cell. For waste products to leave the cell, they also have to go through the cell membrane, and there will be a transport protein to allow them to leave the cell membrane if they need a protein to leave the cell membrane. And if they need a protein to leave the outer membrane of a gram-negative cell, they'll leave through the porin protein. All right? Any questions about any of that? So hopefully you've discovered that the gram-negative cells are different from the gram-positive cells. There's several differences. And the thickness of the peptidoglycan layer is the main difference, but there's also the outer membrane of the cell wall, which is lipid rich. Both phospholipids are found, and lipid A are found in the outer membrane of the gram-negative cell wall. There are few other differences, like the old polysaccharide tail is only found on gram-negative cells. Porin proteins are only found on gram-negative cells. And this is the same picture, just showing you to you in a bigger picture. There is no teichoic acid in the gram-negative cells. They do have, however, a lipoprotein, and that can be connected to the cell membrane and can span the cell wall and can connect into the outer membrane of the cell wall. And that would be a lipoprotein. I don't want to talk about that, so I think that's it. So the gram-negative cell, the outer membrane, has lipopolysaccharides, lipoproteins, and phospholipids in it. Like I said, I'm not going to talk about the periplasm. That's just the space in the cell wall where the peptidoglycan is. The outer membrane of the gram-negative cell can help protect the gram-negative cell from various things, including complement protein and from certain antibiotics, like penicillin G cannot get through the outer membrane of a gram-negative cell, so penicillin G would not be effective on gram-negative cells. You can find some antigens in the outer membrane of the gram-negative cell, and that can give the cells antigenic differences, like E. coli O157H7. These are antigens. O157, the old polysaccharide that comes from the outer membrane of the gram-negative cell. And that's a bad variant of E. coli. It can make people sick. Lipid A is found in gram-negative cells, and it's an endotoxin. So whenever a patient has a gram-negative infection that has a cell wall, the patient will be exposed to endotoxin, and that is always a concern. Any question about any of that? So if you have a gram-negative infection and the cell has a cell wall, then you are always exposed to endotoxin. And that is generally bad, a bad thing. And the patient will have to deal with that. And then the gram-negative cell has the porin proteins, and those are channels through the outer membrane of the gram-negative cell. Any question about any of that? So when we look at the differences between the gram-positive cells and the gram-negative cells, and we're only looking at the differences in the cell wall. The gram-positive cell walls have a thick peptidoglycan. The gram-negatives have a thin peptidoglycan. The gram-positive cell walls have teichoic acid and lipoteichoic acid. The gram-negative cell walls have no teichoic acid. The gram-positive cell walls do not have an outer membrane with them. Did I say that right? The gram-positive cell walls have no outer membrane. The gram-negative cell walls have an outer membrane, and that outer membrane has lipid A with it, and that's important because lipid A is an endotoxin. Any questions there? You guys are all quiet, and I'm not seeing anybody, so hopefully everything's clear. Ah, there we go. I see somebody now. 2 people there. All right. No questions. Let's move on. Let me go ahead and end the show here. Let's talk about the gram stain and how the cells differ, meaning the cell wall gives rise to how the stain stains under the gram stain. So the crystal violet iodine complex forms in the cells, in both the gram-positive and the gram-negative, but in the gram-positive cells, the crystal violet iodine complex do not leave the thick peptidoglycan layer. They get stuck in it, and they do not wash out in the decolorizing step. On the gram-negative cells, the alcohol acetone dissolves the outer membrane because it's lipid rich, and alcohol and acetone just destroy lipids. And then the crystal violet iodine washes out of the thin peptidoglycan layer in the gram-negative cells during the decolorizing step. And so the cells at this step then become clear, and that allows the cells to stain pink when we then apply safranin. Any question about any of that? This slide is showing you a good summary, and you only need to know the top part of the differences between the gram-positive cells and the gram-negative cells. And even here, I can't see it. Let me see if I can blow that up a little. In the gram reaction, there's differences. The gram-positives retain the crystal violet iodine complex, so they will stain blue in the gram stain. The gram-negative cells, let's see if I can move that over a little, can be discolorized in the decolorizing step, and then they accept the safranin stain, so they'll stain pinkish or red in the gram stain. And the peptidoglycan layer is what is responsible for the color difference in the gram stain. The peptidoglycan layer is thick and multi-layered in the gram-positive cells. The peptidoglycan layer is thin. It's not a single layer of peptidoglycan. It's just a thinner layer of peptidoglycan. So that's actually a mistake in the slide. It's not a single layer of peptidoglycan. It's just that the layer is much thinner, and they're meaning that it's a single layer of peptidoglycan. You can find teichoic acid in the gram-positive cells, and they're absent in the gram-negative cells. We're going to skip the periplasmic space because I wasn't discussing it. The outer membrane is absent in the gram-positive cells, it's present in the gram-negative cells. And then the lipopolysaccharide, there's none in the gram-positive cells, and it's found in the gram-negative cells. It's in the outer membrane of the gram-negative cells. And the rest of this you can ignore. All right, any question about any of that? Now that talked about the typical cell walls of the bacteria. There are prokaryotic cells which are atypical, and I should state that the archaea are all, they all differ from the cell walls of the bacteria. So the archaea are in a different domain. They're their own domain. Sorry, I didn't mean to do that. They're their own domain, and they differ from the domain bacteria. Most of the archaea lack cell walls made of peptidoglycan. They do, most of them have cell walls made of pseudomurine or another disaccharide, and some of them even lack cell walls, but most of them do have a cell wall, but it's not made of peptidoglycan. And so the archaea have atypical cell walls, because we're only really going to talk about the cell walls of bacteria. Okay? Now, of the bacteria, there are two families of bacteria that have atypical cell walls. The first family is the mycoplasmas, and that comes from mycoplasma, that family of bacteria cell. These are bacteria cells, but they lack having cell walls. And they do not have cell walls because the mycoplasma are parasites, and they're living with other cells, either plant cells or animal cells, and they're relying on the host to provide them with a suitable environment. Anyways, they lack a cell wall. They do have sterols in their plasma membrane to help give the plasma membrane strength, and that's unusual for bacteria. Sterols are something we find in animal cells, which also give the plasma membrane strength. Okay? Anyways, that's the mycoplasma. They're unusual because this family of bacteria do not have a cell wall. Any question about the mycoplasma? We probably won't discuss them much, if at all, in this class. And if you notice, whenever I'm talking about bacteria and I say something like, if the cell has a cell wall, then such and such, and what I'm doing is I'm qualifying it because the mycoplasma do not have a cell wall. Okay? And if they don't have a cell wall, how do you think they stain in the gram stain? Are they gram positive or are they gram negative?
Gram negative?
Yeah, they stain gram. They stain gram negative because they don't have a cell wall, so the crystal violet iodine complex washes out. But they do stain gram negative. So in that sense, they stain like other bacteria cells. And I guess I should mention that the archaea do have gram-positive cells and gram-negative cells. And believe it or not, the gram-positive archaea, let me show it there, they have a thick, trying to get it in the, there we go. So they have a thick cell wall, and that's why they're staining gram-positive. And then the gram-negative archaea, they have a real thin cell wall, and that's why they stain gram-negative. So the gram stain works on the archaea as well as the bacteria. And it has to do with the same thing, the thickness of the cell walls. The Mycobacterium is in the family, or in the domain bacteria. They are a different family, and they are atypical because they have lots of lipids in their cell wall, and that lipid is mycolic acid. The mycolic acid is why we call them Mycobacterium, the family. Because there's so much lipid, the mycolic acid in the cell wall, they stain poorly, if at all, with the gram stain. And the lipid in the cell wall prevents water from coming across the Mycobacterium cell wall. And the gram stain is water-based. And if water doesn't go across the cell wall, then the gram stain is going to get in the cells poorly, if at all. And so the mycobacterium are atypical because of the mycolic acid. And they stain poorly, if at all, in the gram stain. They are actually considered gram positive because if the gram stain is done correctly, the cells will lightly take up crystal violet and retain it. And their cell walls are thick, so that is true that they should be staining gram positive, but they resist taking up the crystal violet, and so some of the cells will not take it up, and then they'll stain pinkish from the safranin, meaning that they take up a little bit of safranin, but they'll be weakly staining either purplish or pinkish. And so sometimes when you stain the mycobacterium, they will stain variable. Well, some will stain purplish and some will stain pinkish. But if you use the gram stain correctly and you have good staining procedure and you're staining for the full minute and you're removing the majority of the water and you have fresh cells, they should stain lightly purple. Okay? But my point is the mycobacterium are atypical cell walls because they have a lot of lipid in it, and that lipid is mycolic acid. It makes these cells very different because the cell wall can actually protect the cell from, in this case, the crystal violet, but it can also protect the cell wall from other things that are diffusible in water, and that could be various detergents or various antibiotics. There are a lot of antibiotics that are water soluble, and they will not work on the mycobacterium because the cells resist anything that dissolve in water. Does that make sense? And we'll talk more about the mycobacterium later in the term. But for now, just state that they're atypical cell walls because of the mycolic acid in the cell wall. All right, any questions about that? You can damage the cells by damaging the cell walls, and you can do that with actually any of the members of the penicillin family. As I stated, penicillin G gets inside the cell wall when it's forming and prevents the peptide bridges from making cross links in the peptidoglycan. And that's how penicillin and all members of the penicillin family act. They damage the cell wall by keeping it from linking together, and that can result in osmotic lysis of the cell walls. You can also damage prokaryotic cells, at least bacteria cells, by using the enzyme lysozyme. It is an enzyme that digests the disaccharide and peptidoglycan, meaning the NAG and the NAM. And if you expose the cells, the bacteria cells, to lysozyme, you dissolve the cell wall, and then that will result in the cell undergoing osmotic lysis. Where do you think you can find the enzyme lysozyme? Does anyone know? You might have heard about it in A and P. Nobody knows? That's an enzyme that you make to help defend you from bacteria. So you can find the enzyme lysozyme in the cells or on the cells of your skin, as well as in your tear ducts and in the mucous membrane of the mouth and I think the nose. And what it is your body is making it to help defend you against bacteria. In the old days, when a scientist wanted to get a good source of lysozyme, they just collected their tears and then they exposed the bacteria to their tears. So the tears are a good source of it. As I stated, there are differences between the gram-negative bacteria and the gram-positive bacteria. The gram-negative bacteria are not susceptible to penicillin G, whereas the gram-positives are. And the penicillin G just cannot get to the peptidoglycan in the cell wall of a gram-negative cell, meaning the penicillin G cannot get through the outer membrane, and so it does not damage the cell wall of a gram-negative cell. The gram-negative cells are also not susceptible to certain other antibiotics and certain detergents. And I can't tell you the name of the detergent, but let me see, penicillin V is an example. It does not get through a gram-negative cell. Not all members of the penicillin family are that way. Most of the later members of the penicillin family can work on gram-negatives. And ampicillin is one of the early members of the penicillin family, and it works on gram-negative cells. And most of the later penicillin members can work on gram-negatives, as well as working on gram-positives. There are other antibiotics. Obviously, ampicillin would be a member of the penicillin family. Carbapenicillin could work on gram-negative cells. But anyways, there's other antibiotics that can work on gram-negative cells, such as streptomycin, which can penetrate the outer membrane of the cell wall. Streptomycin is lipid soluble, so it would go right through the outer membrane of the cell wall. Any question about that and the damage of gram-negative cells or the protection that the outer membrane gives to gram-negative cell walls from like penicillin G? No questions about any of that? All right, let's briefly talk about the plasma membrane. It's a phospholipid bilayer, meaning there's two phosphates coming together in a lipid bilayer where the fatty acid tails are on the inside because they don't like being exposed to water, and the phosphate portion is on the inside of the cell, on the outside, because there's largely water outside the cell and then largely water inside the cell, and the water inside the cell is in the cytoplasm. Besides the phospholipid bilayer of the cell membrane, there are also proteins, and there's three types of proteins found in the cell membrane or the plasma membrane. Remember, those two terms are the same. Cell membrane is the same as plasma membrane. There's peripheral proteins, and those are proteins found on one side of the cell membrane or the other, but they're not spanning the membrane, okay? They're on one side of the cell membrane and the other. We give it the name peripheral protein. If they span the membrane, they're either an integral protein or a transmembrane protein. Okay, any question about the plasma membrane? Are we at our stopping point? Anybody remember?
Yeah, we're 5 minutes over.
Oh, you guys are supposed to tell me. Okay, let's go ahead and stop it here, and I will see you in the lab at 2:30. Okay? All right, I'll see you later.